When you think of protein, you might think of a big, juicy steak or getting swole at the gym. Fundamentally, proteins are microscopic macromolecules that are vital to organic life. Proteins are responsible for everything from breaking down the food we eat to transporting nutrients from one place to another. We can think of proteins as small biological machines with highly specific functions. The primary or foundational structure of every protein is a string of amino acids. There are 21 biologically produced amino acids, each with different characteristics. The amino acid sequence dictates the eventual structure and function of the protein. If just one amino acid changes, the entire protein could fail to function. The amino acid sequence is almost like computer code telling a computer program how to function. By understanding amino acid sequences, we would be able to code our own protein machines to degrade excess plastic or even fight off cancer. Before making our own proteins, however, we have to first learn how to sequence the unknown amino acids of a protein. Protein sequencing involves two main steps: Edmond degradation and mass spectrometry. First, phenylisothiocyanate, or PITC, is used as a molecular scissor to cut off the first amino acid of the protein sequence. The cut off amino acid is then sent to a mass spectrometer, which acts as a scale that measures mass. Since each amino acid has a different mass, we can identify the amino acid from the determined mass. After that, we just cut off the next amino acid with a PITC and repeat. Seems easy enough. However, there is one major problem. Every time PITC cuts off an amino acid, the protein itself becomes degraded and contaminated. Eventually, after around 50 cuts, the protein is so degraded that any further amino acids that are cut off become inaccurate and unusable. So how do we sequence proteins with upwards of 500 amino acids? The answer is by using more molecular scissors. A class of proteins called proteases are known to cut other proteins at highly specific locations. For example, trypsin always cuts between a lysine and arginine, and pepsin always cuts after a phenylalanine or a tyrosine. By using these proteases to fragment larger proteins, we can then use PITC to sequence the smaller fragments. Together, let's sequence a small protein. To start, we will use PITC to sequence the first few amino acids before the protein is contaminated. Next, since we see a lysine and arginine, we can use trypsin to cut the protein and then continue using PITC. However, we see that the protein has two cut sites for trypsin. Now we need to find out which fragment is which. Let's use another protease such as pepsin to cut another fragment. By overlapping various fragments together, we can assemble the final amino acid sequence of the protein. By sequencing proteins, we can better understand how amino acids determine protein function and one day begin coding our own proteins.